Okay, let's get started. All right, so we talk about physical and chemical control of microbial growth in the last chapter. And this chapter is mainly dedicated to antibiotics because we are pretty much focused on bacteria. And so I call antibiotics chemical warfare because it literally is the chemical warfare between us and bacteria. And, of course, there are other things like fungi involved because the original antibiotics was actually made by fungi. So this is an overview of the chapter. So we start talking about the history of chemotherapy. So most of you guys, if you're like me, when you hear the word chemotherapy, you automatically think about cancer because that is one of the major cancer treatment. But in our context, chemotherapy is much more, it's broader. So it's literally just using chemicals for treating disease. So in our case, we are actually still talking about using antibiotics to treat a bacterial infection. And we'll talk about some fundamental in using antibiotics for treatment. And then two main part of this chapter is the mechanism. So basically, what are the cellular targets for the different antibiotics that we have? And also drug resistance, what's the mechanism behind drug resistance? We will talk about testing effectiveness, basically, Kirby Bauer and MIC. And then a lot of the antibiotics we have today are not working anymore. So we'll briefly talk about our current strategy for discover new antibiotics. So if you think about the history of antibiotics, the first one was discovered, accidentally by what's his face? Alexander. Guys, what's his name again?
Is this Fleming?
Fleming, very good. So by Fleming, and so it was 1928, and then the first antibiotic that was used kind of commercially was actually not until the 1940s. So basically around the time of World War II. And at the time when antibiotics were, the different drugs were being discovered, a lot of scientists were actually being really optimistic and thought that we have found this miracle drug, basically that can kill bacteria. So they thought that eventually our infectious diseases will actually be eradicated. But now, almost 100 years later, infectious disease have not been eradicated, and knowing the nature of infectious disease, it probably never will be eradicated. As a matter of fact, in recent years, WHO has actually sent out a warning saying that we are probably in what we call post-antibiotic era, meaning that a lot of the antibiotics we have been using over the years are no longer effective in treating bacterial infection. So we are kind of in a crisis for the lack of drugs. Because if bacteria become more and more resistant, eventually a simple bacterial infection could actually potentially kill you. I like the jigsaw puzzle because when it comes to using antibiotics for therapy, it's not just a drug, and you actually have a lot of other things to think about. For example, the nature of the bacteria. So what is the bacteria that's causing the infection? What's the site of the infection? And also the patient's health history. All of those actually need to go into consideration. So it's actually a pretty complex picture of which drug to choose for treating what kind of infection. So the main goal for using antibiotics for treating infection, there's this whole table about this is the ideal drug. Of course, this is basically your wish list. So number one is you have to have a pretty good selective toxicity, meaning that you want the drug to be able to kill the microbe, but not toxic to the host cell. And in general, you would prefer the drug to be cyto instead of static, because static, you just simply keep the growth kind of in check. In reality, if the drug is cytal, it might be high dose and it might end up being toxic to host cells. So it's actually, this is the wish list, but in the end, it's kind of a compromise of different criteria. Another consideration is also when drug goes into your body, drug gets metabolized by our body. So you want the drug to be basically in place for long enough so that it can actually have an effect on bacterial growth. And you want the drug to be easily delivered to the site of infection. So if it's if it's just a broken skin, it's very easy to deliver. But if you have a systemic infection, you have to use injection and other methods to deliver the drug. So those are all considerations. So like I said, chemotherapy is literally just using some kind of chemical to treat disease or even prevent disease. So the word prophylaxis literally means using a drug to prevent infection. So before you even get the infection, you can start taking drug. One perfect example of prophylaxis is actually HIV. So high risk group of people can actually take anti-HIV drugs so that can decrease the risk of getting HIV infection. So antibiotics can be categorized based on their origin. So the initial one, penicillin, for example, this is actually the plate that Fleming, so legend has it that he went on a vacation. So he was studying Staphylococcus bacteria, and he went on holiday. And then when he came back, and he saw a plate that has Staphylococcus growing colonies, but then there's also a mold colony that contaminated his plate. It's very observant himself for him to actually observe that around the penicillium, so this is the mold, the fungus, around the colony of the mold, there's no Staphylococcus growth. So instead of being mad that his experiment got contaminated and threw the plate out, he actually thought, well, the mold must be making something that's inhibiting bacterial growth. So this is a replica of his original experiment. Once again, you can see the Staphylococcus growing, and then where the mold is growing, you can actually see this zone of inhibition. So basically, the mold is secreting a chemical to prevent bacteria from growing. So strictly speaking, antibiotics should be natural compound, so such as penicillin, which is secreted by penicillium, which is the fungus. But scientists, humans have taken a natural compound and then start modifying them, so adding a side group of a different functional group. So some of the drugs are actually semi-synthetic, and actually other drugs are completely synthetic, meaning that they don't exist in nature, so we basically synthesize them in the lab. Antibiotics can also be categorized based on the range. So some of them are very broad spectrum, and then other ones are actually narrow spectrum. But the original antibiotic was discovered because fungi and bacteria, if you think about their role in ecosystem, they actually use a lot of the same resource because both of them are actually decomposers. So this phenomenon, antagonism, basically fungi are actually secreting a chemical to inhibit bacterial growth to eliminate competition. So it actually come from them competing for the same resource. So before if a patient walk into your clinic and you basically have to diagnose them and then give them a prescription, these are the things, a few things to consider. So what's the nature of the microorganism? Is it bacteria? Is it virus? Is it fungus? And also what kind of bacteria, if it's a bacterial infection. And also if the if it is a bacteria, is it sensitive to different drugs or is it resistant? Because that's really important. You don't want to give a patient the wrong drug and they take it for 7 days and it doesn't have any effect if the bacteria is very resistant. And also the overall medical condition of the patient. So ideally, you want to know all of these things. You want to be able to actually precisely identify the infectious agent. So it's definitely Staphylococcus aureus and it's not a strep or something else. And then give the targeted the drug to the patient. And for quick identification, a lot of time you can actually start by directly looking at the sample, if it's stool sample or sputum or body fluid and stuff. In reality, a lot of time, because identification take time, doing a susceptibility test also take time. So sometimes physician will actually give the patient a broad spectrum antibiotic to start with. And then once you have identified the microorganism and also know if the microorganism is sensitive or resistant to the drug, you can then switch to a more targeted approach. So for testing susceptibility, a standard method is Kirby-Bauer. So basically, if you were doing this in the lab, which actually we do when we were meeting face-to-face, so you take a plate and then you basically smear a bacteria on the plate. So let's say this is E. coli. So you basically create a smear, a bacterial lung that contains E. coli. And then you put these discs that contain different antibiotics, and then just put them on top of the bacterial lung. And then you incubate the plate and let the bacteria grow. The next day you come in and you use a ruler to measure the zone of inhibition. So if you have a very large zone of inhibition over here, for example, it's good because that means the drug is effective in inhibiting bacterial growth. If you have a very small zone of inhibition, it's not very good. So that means this particular bacteria, so E. coli, is actually resistant to this particular antibiotics. So you do not want to use, to give the patient this particular antibiotics, and you actually want to give them something that's actually still working against the bacteria. So for different drugs, we actually have this chart to show you how to interpret your Kirby-Bauer test. So for bacitracin, for example, if your zone of inhibition is larger than 13 millimeter, so that means the bacteria is still susceptible or sensitive to drug treatment, so you can actually use it. If your zone of inhibition is less than 8 millimeter, the bacteria is resistant, so you do not want to use it. So this is just an example for Staphylococcus aureus. Zone of inhibition ended up being 15, so it's still susceptible. So it's okay to use bacitracin for treating a Staphylococcus aureus infection. This is not the MRSA. This is just your regular garden variety Staphylococcus aureus. So so that's Kirby-Bauer. The downside of Kirby-Bauer is every disc contains one antibiotic and it's a certain dose. So the penicillin, it might be 30 milligram, but you cannot test the dose. So this is where this other test called E-Tex comes in. So E-Tex, same thing, you basically create a bacterial lung. So each plate can contain, either Staph aureus or E. coli or Pseudomonas aeruginosa. And then you have these strips that contain different antibiotics. And the strip actually contains a concentration gradient of each drug. So that's why it's not just a zone of inhibition like a circle. It's actually a different shape. So, over here you have low concentration drug. So the zone of inhibition is actually very, very small. And then as the strip goes outwards, the concentration of the drug increase. And then basically your zone of inhibition also goes larger. So E-test will actually give you some dosage information. And then the other test is the tube dilution, which can be done in this 96-well format, or you can literally just use test tubes for doing different dilution. So the idea is to have different dilution of the drug. So once again, this also gives you some dosage information, which Kirby-Bauer doesn't, because each disc contains just one single concentration. And then you try to inoculate bacteria into the different tubes, and then you look for the minimal inhibition concentration. Basically, what is the lowest concentration of the drug where you do not see bacterial growth? So if you look at the tube, the control, the negative control, does not contain the drug. So after growth, you will see turbidity because the bacteria will actually happily grow. If you have very low concentration of the drug, bacteria will also happily grow. And you are actually looking for the lowest concentration where you do not see turbidity, and that's going to be your minimum inhibitory concentration. Of course, this is an in vitro assay, meaning that we are doing it outside of the body. So the dosage you come out from an in vitro assay does not necessarily correlate with in vivo effect. So you have to take that into consideration also. So what are some reasons that antimicrobial chemotherapy doesn't work? One possibility is maybe the drug cannot get to the site of infection. The brain is a perfect example. So the brain has the BBB, brain-blood barrier. So it's actually notorious for drug to difficult to get to the brain. And then also a lot of time infection is not caused by one single pathogen, right? So bacteria form biofilm and you end up having a mixed infection. So some of the bacteria in the biofilm are sensitive to the drug, but other ones are resistant to the drug. So in the end, taking the drug is not gonna clear the infection. So usually if one drug fails, a physician will give you a different drug, and hopefully by that time they will have more information about the pathogen. And also sometimes they will actually give you a combined therapy. So they might give you a cocktail of more than one antibiotics, or they might change the method of administration to basically help you clear the infection. So so minimum inhibitory concentration, as we mentioned, is basically the minimum dose of the drug that can inhibit bacterial growth, right? So if you basically look at a curve, for example, over here, so when you increase the dosage of the drug, the bacteria will basically, hopefully, the growth will slow down and eventually they don't grow at all. So that's MIC, minimum inhibitory concentration. When we think about a drug, it's really important to think about selective toxicity, and selective toxicity is actually being measured by this parameter called therapeutic index. So therapeutic index is literally the toxic dose divided by minimum inhibitory concentration. So it's actually considering what is the concentration that's effective in killing bacteria, and then what is the dosage that can actually causing toxic effect on the patient. So if you look at this curve, for example, so here is the therapeutic dose, and then here is the toxic dose. So if the two curves are very far apart from each other, meaning that, so let's say penicillin. So 10 milligram is very effective in killing bacteria. And then if you take a penicillin that's 100 milligram, then you can see some side effect. So you end up having a therapeutic index of 10. Then it is a pretty safe choice, because if 10 milligram of penicillin does not clear the infection, you can actually play around with the dosage. You can increase it to 20, you can even increase it to 30, knowing that, when you increase it to 100, it started having side effects. So you can actually play around with the dosage. On the other hand, if it's a different drug where the TI is 1.1, so basically 10 milligram, is barely able to deter bacterial growth. And then if you increase the dose to 11 milligram, the patients start showing side effect. So then it's a really risky choice, because you don't really have a lot of room to play around with the dosage. So usually, ideally, you want to find the drug that has the highest TI, because you have wider margin of safety. So this is kind of repeating some of the information we have just talked about. So once again, when it comes to drug choice, you really have to think about a lot of the things, including the patient's physical health, especially if they have liver and kidney disease. That kind of limit your choice, because liver and kidney are involved in metabolism of drugs and stuff. So if the patient's liver or kidney don't function very well, that kind of limit your choice. Also the patient's age, whether they're pregnant or not, and do they have some kind of metabolic abnormality, and what kind of other drugs they're also taking, because drugs have a tendency to interact with each other. And then, of course, there's also how do you administer the drug and the cost of the drug. So this is actually a case study that came from the previous textbook we're using, but I actually really like it. So that's why I keep using it, and I like to talk about it in the class. So basically the case is you have a patient that walks in who's elderly, who is also an alcoholic, and he's presenting, basically he has pneumonia, so he has inflammation in the lungs, and you have already identified the causative agent, the pathogen is Klebsiella. And this is what else you know about the patient. So the patient actually does have liver and kidney dysfunction. So you have to take that into consideration. So you do, let's say, Kirby-Bauer or the E-test or the tube dilution test, and you figure out that the particular strain of Klebsiella is sensitive to these three drugs, so cephalosporin, gentamicin, and imipenem. So basically, you have these three to choose from. But first of all, cephalosporin, it is known that it have a tendency to interact with alcohol. And for patients who's alcoholics, it can actually cause, especially patients who are elderly, it can actually cause internal bleeding. So that's pretty severe side effect you do not want to see. So cephalosporin is out. Gentamicin belong to aminoglycoside, so these are protein synthesis inhibitors. But gentamicin is also known to be pretty toxic to the kidneys. The patient already have a kidney problem, so you do not want to use gentamicin. So that basically leave you with this one drug, imipenem. And imipenem is not perfect, so it's known for patients to take imipenem, they end up having stomach problem or intestine discomfort. But if you look at the big picture, it's definitely not as toxic as gentamicin or cephalosporin. So in this case, imipenem is actually going to be your best choice, and that's probably the drug that you are going to prescribe to this particular patient. So once again, you have to take into consideration a lot of the factors before. And then today, there are actually apps on smartphones to help doctors to choose the right drug to give patient. Okay, so let's talk about the cellular drugs, cellular targets. We talk about selective toxicity. So basically, you want the drug to target the microorganism to kill them or prevent them from growing, but you don't want them to cause side effects. So basically, you don't want the drug to target your own cell. When we come to, so penicillin, for example, has a very large TI, a therapeutic index. So it's usually pretty safe to play around with the dosage. When it comes to cellular target, there are 5 classes of antibiotics. So penicillin belongs to cell wall inhibitors. There's also a very large class that inhibit protein synthesis. So basically, the drugs will target ribosomes. And then some drugs can also target DNA and RNA synthesis. Other drugs can actually target the cell membrane. And then the last class is the class that target folic acid synthesis. So we kind of generically talk about these drugs as metabolism inhibitors. And then this is just an overview, basically repeating the information. You can see that, so there's actually a whole bunch of drugs that target cell wall, including penicillin, cephalosporin, we just talked about. And actually both of them belong to beta-lactam drugs. We'll talk about beta-lactam in a minute. And then we have drugs that target the ribosome. So remember, ribosomes have a large subunit in bacteria, that's the 50S subunit. And then the small subunit is the 30S subunit. So different drugs will actually target either the small subunit or the large subunit. Some drugs target DNA synthesis, so enzymes that are involved in DNA replication, or enzymes that are involved in transcription, so RNA synthesis. And then other ones target membrane, so polymyxin target membrane integrity. And then the metabolic pathway, we're talking about folic acid synthesis. So sulfur drugs and trimethoprim belong to this particular class. So let's talk about cell wall inhibitors first. So penicillin, remember, this is the first antibiotics ever identified and purified. So this is the structure of penicillin. So in the middle of the chemical penicillin, there is this beta-lactam ring. So for penicillin to be able to basically inhibit bacterial growth, this beta-lactam ring has to be intact. How does penicillin kill bacteria or prevent them from growing? It basically can bind to an enzyme in bacteria called transpeptidase. So if you guys remember, the cell wall of bacteria are made of peptidoglycans. So a lot of the long sugar chains cross-linked by peptides, so amino acids form peptides. So when penicillin binds to this transpeptidase, inhibit its activity, the peptidoglycan cross-linking does not happen. So then cell wall synthesis is basically inhibited. So because transpeptidase is where penicillin can bind, there's another name for the enzyme, which is called penicillin binding protein. So PBP and transpeptidase is pretty much the same thing. So PBP, transpeptidase describe its activity to cross-link, and then PBP just describe that it's capable of binding penicillin. So the initial penicillin, that was the first generation of penicillin, were actually natural compound from fungi. And so penicillin G and V, you can actually see that. So for the most part, structurally, they're very similar. The only difference is in this R group. So with G, you just have the hydroxy group. With V, you actually have a different side group. And they are relatively narrow spectrum, meaning that they're mostly working against gram-positive bacteria very effectively. That makes sense because gram-positive bacteria have a very thick peptidoglycan layer. If you inhibit the cross-link, you are going to damage those cells. They can work for a few gram-negative, but not very well. In terms of delivery, penicillin G was parenteral, so basically only through injection. Penicillin V is a slightly improved version. It's oral because in general, patients do not enjoy being injected. A lot of people don't like needle, so oral is a little bit better. It's easier to administer. So once again, this is the first generation. And then scientists, chemists, actually a lot of pharmaceutical companies literally hire a lot of chemists to do this kind of stuff. So they actually start modifying the R group. And that gave us ampicillin and also amoxicillin. So ampicillin and amoxicillin belong to the second generation of cell wall inhibitors. And the modifying of the side group basically increased the spectrum. So now, in addition to working against gram-positive bacteria, these two are actually working for against a lot more of gram-negative bacteria as well. In terms of the delivery, once again for ampicillin, either needle injection or oral. For amoxicillin, it's oral. So that gives you a better drug that can target gram-negative bacteria as well. But then, by that time, we are already seeing bacteria that are resistant to penicillin. So methicillin is actually designed basically for that purpose. So it's also semi-synthetic, basically taking the structure of penicillin and modifying the R group. But it is used for treating bacteria that are already resistant to penicillin. In terms of delivery, it's also injection. So it's actually the third generation. Basically, if penicillin doesn't work, you might give patient methicillin because the R group is more bulky, so it's harder for bacteria actually start producing this enzyme called penicillinase or beta-lactamase. They can literally cut open the beta-lactam, and that's why the original penicillin doesn't work. But this methicillin, because the R group is bulky, it's harder for the enzyme to get to the beta-lactam ring. So it still work for bacteria that are resistant to penicillin. Unfortunately, you guys know about MRSA. So MRSA is methicillin resistant staph aureus. So we now have bacteria that are resistant to methicillin as well. So this is just a slide showing you guys penicillinase, which is also called beta-lactamase. So this is an enzyme that some bacteria can produce. So basically, the enzyme can cut open the beta-lactam ring of penicillin and actually any drug, ampicillin or amoxicillin, any drug that depend on the integrity of the beta-lactam ring. If the bacteria can produce an enzyme to cut open that ring, the drug is not going to be very effective. So penicillinase basically means that it can make penicillin not very effective, so it can destroy penicillin. Beta-lactamase, same thing, it basically target the beta-lactam ring and cut open the beta-lactam ring. So the two of them are actually pretty much the same thing. So that actually leads us to the fourth generation of drugs. So basically, the fourth generation is also further modifying of the side group, basically make them even more broad spectrum. So broad spectrum are useful in the sense that when you don't know what's causing the infection, you can give a patient a very extended spectrum, and hopefully that can temper down the infection. A more recent discovery is also this chemical compound called clavulanic acid. So clavulanic acid is actually a beta-lactamase inhibitor. So remember, beta-lactamase is the enzyme that bacteria produce that make penicillin useless. So we have discovered an inhibitor for the beta-lactamase. And then if you combine penicillin this clavulanic acid, basically, even if the bacteria have a resistant mechanism to produce beta-lactamase, because the drug contains this chemical that can make the beta-lactamase ineffective, penicillin can still kill the bacteria. So this is what I mean by chemical warfare. We are literally waging a chemical warfare with bacteria. We use drug to kill them. They come out as a resistance mechanism. We end up discovering newer drugs, or we end up discovering inhibitor of their resistance mechanism. So it's kind of an ongoing chemical warfare between humankind and bacteria kind. So that's penicillin, so that family of cell wall inhibitor. Another large family of cell wall inhibitor are cephalosporins. So the initial cephalosporin, the first generation, is also a natural compound coming from the fungus cephalosporium. The story is actually really similar to penicillin. So if you look at the chemical structure of cephalosporin versus penicillin, it's also a beta-lactam drug because it also have the beta-lactam ring. The first generation mostly target gram-positive bacteria. Second generation, more broad spectrum, so you can actually use them against gram-negative bacteria. The third generation, also broad spectrum, actually really effective in treating enteric bacteria. So these are bacteria that live in our gut that can actually also cause infection. And then the fifth generation, we're actually now targeting bacteria such as MRSA. So that's cell wall. So cell wall inhibitors tend to have a higher therapeutic index because if you look at their targeting, they're targeting enzymes involved in peptidoglycan synthesis. And in our body, we do not even have that chemical compound. We don't have peptidoglycan. We don't have enzymes involved in peptidoglycan. So you can actually use higher dose and not causing a lot of side effects. Another large class of antibiotics are protein synthesis inhibitor. So aminoglycosides. So these are chemicals so that include gentamicin. So we kind of talked about it early on. Also neomycin, kanamycin, and streptomycin. So they basically target a small subunit of ribosome. So if you guys remember when we were talking about translation, the first thing that happens is the small subunit binds to the messenger RNA, and then the small subunit scan for AUG, the start codon, and then once the start codon is found, they recruit a large subunit to come in, and then once that's done, basically the tRNA will bring in each amino acid, and then you start synthesizing a polypeptide chain. So the A side of the ribosome is where the tRNA comes in, and usually whatever the codon is, the tRNA has to carry the anticodon that's an exact match, right? So if the codon says CGC, the anticodon should be GCG. So what this class of drug, aminoglycosides, does is it actually causes this mismatch over here. So instead of GCG, another tRNA that carries the anticodon GAG is now kind of close enough, but because it actually carries the wrong anticodon, it brings in the wrong amino acid. So we end up synthesizing a protein that contains a lot of mistakes, and it may or may not function, and that's how it can actually kill the bacteria, or at least prevent them from multiplying. So it causes basically tRNA mismatch, and then it causes the wrong amino acid to be incorporated into the protein, and it tends to be cidal, so it can cause bacterial death. The problem with aminoglycosides is actually it is really toxic to a few organs. So it's really toxic to kidney, and it's also really toxic to the ear. So there are actually patients who end up becoming deaf after taking doses of aminoglycoside. So you really have to be careful. And also it causes other neurological problems like loss of balance, and nausea, and also some patients have allergic response to the drug. Another drug that also target the small subunit is actually tetracycline, but tetracycline has a slightly different mechanism. So when tetracycline target the ribosome, it literally prevent any incoming tRNA to go into the A site. So it basically causes premature termination of protein synthesis, and you end up having a protein that's synthesized probably halfway through, and then the bacteria cell do not have the proteins that they need. So tetracycline is very broad spectrum, and it can be used for targeting a whole bunch of bacteria, and they tend to be bacteriostatic, so it's not as lethal, but definitely prevent them from multiplying. The problem with tetracycline, so it was actually pretty widely used back in the days, I think maybe the 60s and something. And then they actually figured out that after people were taking tetracycline, a lot of their teeth actually turning yellow. So that actually is a pretty significant and noticeable side effect. So that's why tetracycline is not being used as widely now. It can also be toxic to the liver. So it is still being used, and also of course there are other tetracycline derived drugs like doxycycline that have lower side effect. So it's still being used. It's just not used nearly as widely as at some point. And then we have macrolides. Macrolides as a class of drug, they actually target the large subunit, or the 50S subunit of ribosome. So if you guys remember, the catalyst of peptides formation is actually that function is mediated by the 50S subunit. So when macrolides binds to the ribosome, it literally prevents peptide bond formation between the incoming amino acid with the previous polypeptide. So it also kind of causes the premature protein synthesis, and you end up synthesizing proteins that are maybe only halfway, or only having a few amino acids. The original macrolides, erythromycin, was a natural compound, so it actually came from Streptomyces. And then we now have other ones that are semi-synthetic, which end up having broader spectrum, and also the chemical modification can increase the shelf life, and also be less toxic or less side effect. So those are protein synthesis inhibitors, and they also tend to have decent therapeutic index because bacteria ribosome are different from our ribosome. So that's why you actually have a whole lot of drugs that belong to the protein synthesis inhibitor. The metabolic inhibitors, so we are mainly talking about the folic acid synthesis. So folic acid is really important in bacteria, and also actually in our cells, because folic acid can be the precursor for thymidine, for purine, for methionine. So you can actually see that it's required for amino acid synthesis, and it's required for nucleotide synthesis. So it's basically feeds into protein synthesis and DNA and RNA synthesis. So the drugs, the two best drugs are sulfonamide, sulfonamides, which a lot of times we just call them sulfur drugs. So sulfur is structurally very similar to this chemical compound called PABA, or p-amino benzoic acid. So PABA is the substrate for the first step in the folic acid synthesis pathway. So the enzyme is called tyrosine synthetase. And because sulfur is chemically very similar to PABA, it can actually act as a competitive inhibitor of this enzyme. So when you have the sulfur drug added, basically this step is blocked, and then you do not have dihydroteric acid or dihydrofolic acid synthesis happening. The other drug, trimethoprim or TMP, is actually structurally very similar with dihydrofolic acid. So it can actually act as a competitive inhibitor for this enzyme, dihydrofolic reductase. So when TMP is present, this step of the metabolic pathway is blocked. You end up not producing tetrahydrofolic acid. So both of them are working on the same pathway. They're just working on different steps of the pathway. And both of them are relatively broad spectrum. The really neat thing about these two is that you can actually combine sulfur with TMP, and they actually have a synergistic effect. So synergistic basically means that the effect is higher than the additive effect. So if a sulfur drug, the effect is tenfold, TMP is another tenfold. When you put the two together, it's not 20, it ends up being 100, because somehow combining the two is just really effective in shutting down the entire pathway. The sulfur and TMP, so these metabolic antagonists actually have a very high therapeutic index, and that's because even though folic acid is also required in our body, our cells actually do not synthesize folic acid. We actually take in folic acid from our diet. So this is why, pregnant women, for example, a lot of the time, doctors will actually caution you to make sure you have enough folic acid in your diet, because we don't have this pathway to synthesize folic acid. We, actually get the folic acid from our diet. So that's why the drug that target this pathway does not have a lot of effect on our body. So, the drug end up having pretty high therapeutic index. And then we have nucleic acid synthase inhibitor. So quinolones, basically they target topoisomerase. If you guys remember from our conversation about DNA replication, so helicase comes in and then start unwinding DNA, and the unwinding, both directions end up creating a lot of tension in the double helix. So we have this other enzyme called topoisomerase that comes in, and basically they can nick open the DNA, one DNA strand, and then release some of that tension. So quinolones literally target DNA gyrase, which is one of the DNA topoisomerase. And then by making the topoisomerase or gyrase not functioning very well, it inhibit DNA synthesis. And the quinolones are actually very effective in targeting enteric bacteria. So they can be used for treating bacterial infection caused by enterics. And then finally, we have membrane disruptor. So polymyxin B. If you look at the chemical structure of polymyxin B, not the chemical structure, but basically the shape, it looks very similar to detergent or surfactant. So basically, it has a long hydrocarbon chain, so a long hydrophobic tail, and then kind of a charged or hydrophilic head. So because structurally it's very similar to phospholipids, it can insert itself into the phospholipid membrane and then disrupt the membrane integrity, and then that can actually cause the bacterial cell to die. The problem with polymyxin B is the bacterial cell membrane is really similar to our cell membrane. So if the drug can disrupt the bacterial cell membrane. It can also disrupt our cell membrane. So that's why polymyxin B is not used orally, in general. It's actually used in, it's one of the ingredients in Neosporin. So that's a topical cream. If you look at the ingredient of Neosporin, it contains neomycin, polymyxin B, and also bacitracin. So it's actually 3 antibiotics combined. So when used topically, it doesn't cause nearly as much toxicity for your body. Polymyxin B tend to be narrow spectrum because it's much more effective for gram-negative bacteria. That also makes sense because gram-negative bacteria have a lot more membrane material. So they have this cell membrane, they also have the outer membrane. But the limitation of it is it's like the toxicity is very poor, and it tend to cause kidney damage and tend to cause nervous system damage. So this is just a chart showing you, in general, we talk about antibiotics can be distinguished based on their spectrum. So you can see that there are some really extreme cases. Tetracycline is very broad spectrum, and then this guy over here, polymyxin, is very narrow spectrum, only for gram-negative bacteria. And then you have other ones that are kind of somewhere in between. This other drug, isoniazid, is also very narrow spectrum because it actually inhibit glycolic acid synthesis. So that's why this particular drug is only used for treating diseases like TB and not for any other bacterial infection. Okay, so here's your first Kahoot question. Okay, not bad for the first one. We actually have 29 people giving the correct answer. So it's a definition question. Basically, what do aminoglycosides target? And the correct answer, of course, is a target a small subunit of ribosome. It's not a cell wall inhibitor. It's not a metabolic inhibitor, and it's not a cell membrane disruptor. Okay, so the success rate for this one is slightly lower. So this one is asking, we have 4 drugs, which one is now targeting protein synthesis? So, this chapter does have a lot of information. Just looking at this one chart, you can be overwhelmed with the information because there's so many drug names and so many different mechanisms. So the key to approach this question it's not necessarily, memorizing that, all of these guys are protein synthesis inhibitor, although, eventually you probably will get there. It's actually if you can spot the odd one out. And you should actually know that sulfur, remember, they're folic acid synthesis inhibitor. So it definitely is not a protein synthesis inhibitor. So that's how I would approach this particular question. Let's see who's on the leaderboard. So we have Quincy, Cynthia, Angeline, Pearl, and Yuanfen on the leaderboard. Good job. Okay, so let's talk about antibiotic resistance. So drug resistance, evolutionarily speaking, it happened way before we discovered antibiotics. So remember, bacteria and fungi have engaged in this chemical warfare with each other for a long time. So resistance of to penicillin probably existed in bacterial population before we even discovered penicillin. But drug resistant, so bacteria that are resistant to penicillin, start being noticed in the 40s. Remember, the drugs were being used starting in the 40s. Actually, some people actually thought that during World War II, because the British had penicillin and the German did not, that actually kind of contributed to the Allies winning the war, and Germans lost it. But yeah, so that actually help you to remember the timeline. But pretty much as soon as we start using penicillin kind of in a wide the population, we start noticing there are some bacteria that are resistant to the drug. By the 80s and 90s, they start seeing a lot of treatment failure. So now we're seeing drug resistance in a much larger scale. And I just really I'm not necessarily a Star Wars fan, but I just thought that it's really, really cute, the resistant movement. So how does drug resistance develop? So in general, come from mutation. So if the drug resistant gene can be either carried on the chromosome or on the plasmid. If it's on the chromosome, most likely it came from spontaneous mutation, a copying error of a kind. If it's carried on the plasmid, remember bacteria do have horizontal gene transfer. And so it's very easy for them to get the plasmid from another bacteria, and through conjugation, through transformation, and to transduction. This cartoon is kind of pretty funny. You have a guy, a bacteria walking, along the alley, and the guy said, "Hey kids, want to be a superbug? Here is a plasmid DNA. Basically, you can stick it to your genome, and then you're resistant to antibiotic treatment." So what are some mechanisms for antibiotic resistance? So there are actually many. Some of them, they can actually decrease, so they can modify their own membrane, and also membrane proteins, so that the drug cannot get into the cell. Or they can modify other target on the cell membrane so that the drug can actually be bounced off. So once again, decrease the uptake. Other ones can actually start making these efflux pumps. So these are really effective because even if the drug gets into the cell, the pump will actively pump them outside the cell. And if the target cannot target the enzyme inside the cell, the drug is not effective. Other ones can inactivate the enzymes that the drug target, or some of them can actually use an alternative pathway. And then some of them can actually change the target, and other ones will actually block the pathway. So there are multiple ways for bacteria to be resistant to the drug. And ooh, that's not good. Can you guys still hear me okay?